Welcome, Forrest, to the Collaborative Research Center Origin and Function of Metaorganisms. It's really fantastic to have you here. You are one of the authorities of viromics and viruses and ecosystems. Maybe can you tell us how did you get to viruses at a time when, now we have technology to detect them, but I assume when you started this research, those were very early days. What did you get into that field?
Yeah, so I was going to be a lawyer. Okay. So I went to grad school and I did do virus research, but like conventional, like on a virus very closely related to HIV. And I was studying HIV in humans, or this HTLV1 in humans. And I had learned a lot of techniques, molecular techniques. And then just before I was going to law school, I saw a talk by someone named Farouk Azam, who is one of the godfathers of marine microbiology. And he told this fascinating story about all these viruses in the sea that nobody knew anything about. And I was like, well, I actually know how to study those because of my previous experiences doing human immunology. So I dropped my law school plans and I went to the Scripps Institution of Oceanography, and we stole some money from one grant to pay for some sequencing, and that's how we did it.
Okay. How
yeah.
Wonderful. Okay.
Yeah.
And so, I assume you never regretted a single day not to go to law school, but doing what you do now.
Yeah. No, I'm glad I didn't go to law school now.
What was the initial insights, observations in the early days from your PhD and from your postdoc work?
So the main thing, which continues to this day, is that the diversity in the viruses is just incredible. We really haven't sampled it at all, even after maybe 15 years now of sequencing and looking in there. We're every time we look, we find completely new viruses, completely new genes, and no idea what they're doing. So it's very much this discovery phase still.
Okay.
Yeah.
So when people think about, or when I tell, or you'd probably talk to people and talk that you're working on viruses, most people consider them as pathogens and bad guys. But clearly, they are part of our ecosystem, and there must be also some maybe even beneficial sides. Do you have evidence for this positive or fitness-promoting effects of viruses or phages?
Yeah. So you get both, right? As there's always the double-edged sword side of it, but definitely the phage in particular can protect animals against bacteria that might be harmful. We see different things where the phage cause recycling of nutrients that people need in both in the ecosystem and in their own body. So there's lots of examples. Even the viruses that we would say are harmful often are not. They're just under certain conditions they're harmful. So it does look like for human immunology, you need a lot of your viruses. And if you don't have them, you run into a lot of problems. Shingles would be like the most famous one that most people do. If you don't already have the Epstein-Barr virus, and you get exposed to it later on in life, you actually end up with a much worse disease. Mm-hmm. So it's better to carry the virus.
Right. Yeah, and so virulence or pathogenicity seems to be context dependent.
It's completely context dependent. Yeah.
Let's go back a little bit to your. You are well known as an expert in measuring, describing, finding diversity, and that needs nowadays a very special technology to do it. Maybe you tell us a little bit about that. And then we are very much concerned about the loss of diversity. And I want to ask you, do you see that also in the viral world? When you sample at certain places, are they more diverse than others? Is there a human factor in influencing viral diversity?
So yes. So the main way that we look at diversity in viruses is that we would go into a sample where whatever environment you want, and we separate the viruses away from all of the cells and everything else there. And then we just use DNA sequencing to look at often billions of different pieces of them. And from that, we can reconstruct how many viruses are there and what types they are, things of that nature. What we do see is that when you go into systems where humans have changed things dramatically, mostly by removing big organisms, you see a really big decline in both bacterial diversity and in viral diversity. So that is a clear indication of something is messed up in the system. Most ecosystems are highly diverse, and when they're going through some bottleneck, they become less diverse.
Would you consider viruses as drivers of diversity?
Yes, they're one of the main drivers of diversity because they actually, of course, select for things that can't get infected by them, which is one, as everybody knows, you could, if you get a little sick and the wrong virus gets you, you're dead. So that causes diversification. But the other thing is that they they move genes around all over the world. And that movement of genes is the main diversity between different organisms, whether it's big things like us or bacteria.
Mm-hmm. One of your other interests, and maybe your heart is in the sea and the marine environment and in the corals. I mean, most of your pictures which we can find are as a diver or a scuba diver, corals, coral ecosystems, also in danger right now. Where is your concern? Where is your interest with the corals? What do you want to understand, and how do viruses fit into that?
Right. So the coral reefs have been the most important place for us to look because we've been able to really understand how viruses contribute to the decline of coral reefs and then apply that to human biology. So what we know is that the main thing that happened to coral reefs was overfishing. So we've been overfishing coral reefs for about 80, 90 years, ever since the end of the Second World War. And the removal of the big organisms off the coral reefs really led to a switch where we have a lot more microbes in those systems. So coral reefs have a lot more microbes than they should. And then those microbes come back and they kill off the corals. In this case, the viruses that infect the bacteria are really the problems because they're bringing along these genes. I told you they can move genes. They move genes into the bacteria, and then and they do it for a completely different reason than killing corals. But the end effect is that they kill corals. And so you get the decline of the coral reef because of this. So that's one of my main things that I look at. I'm also interested in how you might use them to reverse this effect. So right now we're looking very much into restoration of coral reefs, not just why are they falling apart, but is there some way to reverse it?
Mm-hmm. Okay, to go a little deeper in that. So first, I understand that there is a kind of a really negative feedback. If I guess environment changes, then viruses are coming, then they somehow change the microbial community to the worse, and that again has adverse effects on the corals. And coral bleaching is one of the major problems these days. And the Great Barrier Reef, I don't know, 60-70% of the corals are bleaching. But what's your idea of rescuing that or reversing that? Can you tell us a little bit about that?
Well, there's a lot of different ideas.
Okay. Well, if anybody can do something that's not only very important, I guess this is spectacular. I mean, can you share some ideas with us how to go ahead?
Right. So one is, of course, just knowing what the causes are, allow people to really think about how they might regulate for conservation purposes. But given the changes that you're talking about, the global changes, not just the local changes, we're really wondering or trying to come up with systems where we can move whole coral reefs to different parts of the ocean. So we want to move where there's not going to be the increasing water temperature. There's not going to be the decreasing pH, which are the global effects that the coral reefs are really experiencing. And how would you actually move all of the diversity of a reef from one area to another? And that's going to take the viruses to help. Yeah.
So to understand that, one idea would be that you move then a sick reef to a place where it's more protected, and to let it grow again, and maybe at some point then move it back.
That's exactly the idea. Yeah. So move the whole system, restore systems based on diversity that you put in other places. Yeah, that's exactly what we're trying to do now.
Right.
Yeah.
So if people think about phages and viruses, and now rescuing or treating diseases, immediately, of course, human situation comes into mind, and your lab and your team is also looking to some extent also in human disease, in cystic fibrosis, as far as I know. How does that fit into this more ecological and basic research coral type of research? And in the medical era, of course, phages are well known since 100 years, particularly from the Russian literature, as phage therapies. So can you allow us to go with you in your lab, in your more medical-oriented projects, and then tell us your opinion on phage therapy yesterday and today and maybe tomorrow?
Right. Yeah, so that's a great question. So the idea being, could we use the phage to control the bacteria in certain diseases? So again, it was like before, there are lots of double-edged swords. So sometimes the viruses are the real problem, and sometimes they're the real solution. So in cystic fibrosis, we've had cases where the reason that a patient dies is because of a gene brought in by the phage. In other cases, we've been using phage to try to control bacterial infections, and sometimes it works really well. But we need a lot more basic research, because it's the thing about phage therapy and using phage to kill off bacteria is that it doesn't work every time you try it, right? You actually need the rules, and we don't understand the rules. And a lot of the ways that we know that bacteria interact with the phage and the way that they interact with humans comes from basic research in corals and in hydra and stuff like that. So that's really where we're going to figure these things out and really apply them. The future is going to definitely use phage to kill off bacteria. And right now it's getting really exciting, because I can take someone who's sick with some disease that nobody knows about. We can sequence the DNA and figure out what phage are in that system and are killing a particular bacteria. And we can make those in the lab and put them back into the person. And the hope is to do that in a matter of a couple days, instead of right now it takes years, or not years, but weeks to do any of that stuff. Yeah.
Kind of auto-transplantation.
Exactly.
Yeah. Oh, wonderful.
Yeah, you can imagine just taking all the DNA sequence and saying, well, this phage kills this bacteria, and just synthesize it in the lab and treat with it.
Yeah,
so
amazing.
It'd be real personalized medicine.
Yeah. That's an excellent point of changing a little bit the subject from the basic research done in your lab to the junior generation of researchers, which are maybe entering our fields and our labs. This aspect, I want to go to two aspects. One is the communication aspect, how to communicate these findings, but the other, let's start with this perspective which you have now outlined, of course, is fantastic as for a young generation to think of startup companies, of applying that. And would you encourage young people to think about that? I mean, when we talk about bacteria, there is this probiotic field, which is very similar to the phages. And sometimes it works, but nobody knows exactly how it works. So one needs basic research to find that out. But I think in future there is tremendous possibilities for junior people now to really go also in this more, found a company, make a startup, and make use of that. What do you think about that?
Yeah, definitely. No, I agree 100%. Is that there's the economies that are going to come out of both the phage and the bacteria, and just how we manipulate and think about ecosystems is going to be gigantic. And there's a lot of money to be made. And from the point of view of, you don't even have to be young, old. Okay. I consider sometimes starting a company.
Okay.
But it is like it's a wonderful place to be working, because there's it's also it's expanding so much that you can start in kind of an obvious place and you'll end up in all these non-obvious places. So agriculture, obvious place to start because people want to do it. But that's leading to a whole bunch of other areas that people never thought of using these things. So,
Yeah, excellent. And one more sentence from my side, but what I find fascinating is that there are excellent, very old work. I mean, goes back to Mechnikov and the Russians, etc. They did very careful observations, but of course, these were just on the surface. But they had this good gut feeling that something is really going right. And the chances today are that one can find the mechanisms and then the molecules and the substances, and then you know what you're doing, and then you can go really to treatments. And so, very nice. Science communication, a big challenge these days. I mean, if you talk to somebody about viromics, I mean, who knows what does he talk about? What is viromics? If I talk to somebody about meta-organism or holobiont, what is that? So, in our view, it needs concentrated efforts and intelligent efforts to communicate this tremendous, useful, exciting, important, basic research observations to the general public, but also, to maybe even scholars in academia, which just do not understand us because we talk a language which they do not understand. But if you want to really do interdisciplinary work, then we have to talk in a language where we reach each other. How are you doing this?
Well, it's very hard. It is, so what I'm finding, or what I think works the best, is some sort of fusion of the science with the arts, because people really respond to that, right? So it's got the emotional side, and then they can get into learning some of the details about the science. Going directly with science, it's like all of us, right? When you go home, you don't necessarily want to just read a scientific paper. You probably want to watch some fun movie or listen to some music. And so that's, of course, in the free time, you're competing for people's interest in the free time. But what I do find is that if you get some emotional appeal to people, they will learn very detailed stuff.
Can you give us an example where you use the arts to communicate some of your findings? Which type of arts? Painting, music, whatever. And from your side, how that worked well?
Yeah. So you can do it with fine art, actually. So if you can get fine artists to work on it, they do an amazing job, actually. Paintings and things of that nature are very successful to a subset of people, right? But I've actually found that you're much happier with things that are less, more like the graphic novel-ish sort of things, or a little bit more pop culture-ish, because you're trying to get people really excited about it. And that can range everything from music and how music might contribute to someone's understanding of of science, which we do, because you can get a composer to do fun things with DNA sequences or data in general. But the other thing that really seems to work, I think, is something that's visual. So make a very good comic would be my best guess. We do some graphic, like, what do you call it? Graphic design. I'm not doing a good job. Like little films and stuff of that nature. But that's hard, I think, to get people, because there's so much content out there. But people really will latch on to a good picture. Yeah. And you can see things that, like, I've had people make like kind of iconic things that convey something about phage, for example. And you'll see them all over the world now, where people have picked them up and are using them. So that works, I think. Yeah.
Okay, yeah. I mean, your 2 books, which were published in the last few years, all of them have wonderful covers and also graphics inside.
Yeah. Look for people like that, because you actually want the artist that you're looking for. Like, so one of them was a tattoo artist, and then actually did a whole bunch of tattoos of phages because people really wanted it. Like, we had a couple people fly to San Diego to get tattoos of phage because they liked his art so much. And I mean, there's things like that. Yeah.
Do you have any evidence, objective, direct evidence that this really is transporting the message you want? Or, I mean, of course, I find it appealing, and nice pictures are nice, but do we really know that works?
Yeah, so that's really hard. The assessments and really understanding, do people get it? So yes, but frequently what you get is, especially like with viruses and phage, people will go, oh, those are beautiful. I really like them. And that's about where it stops. So getting into the real details is very hard for people. It's not, people are really, almost everybody is extremely smart, and you can get them to get excited about things. But it is, how much time do they have to learn it? And some of the stuff that we do is very complicated. So anything that makes it more simple, even for us, because we're bombarded with so much science ourselves that we run into where we don't think about certain fields because they're just too complicated from our point of view too.
Oh yeah. I totally agree. I think it's exactly also for us ourselves that we can think beyond certain borders without getting drowned into details. We need this help, and which is a kind of a simplification, but it must be still precise and correct. Otherwise, it's useless. Yeah.
So one thing I found is that in the teaching environment, so not just like to the public outreach, but in the teaching environment, is we've set up classes where we have scientists working with artists. And then they have to put together a project. And the project has to be kind of to display some complicated scientific idea. And that's been great for both the scientists and the artists, because they really do exchange. And it is the best way, I think, to teach the artists some really hardcore science. And it really does help the scientists not be so bad at communicating their work, which is, and it's fun. It's actually more fun to teach than most things that you would teach.
Yeah, excellent. We have here an art school in Kiel, and we have similar experiences. And it's fun, and B, it's quite informative and also helpful in terms of supporting interdisciplinary thinking and also raising interest in the subject, of course. Yeah. So art is important. Yeah. I learned that beyond the fine arts and the paintings and the comics and the videos, which we have touched now, music. And I learned that by looking into phage genomes, music can be composed, and you got inspired, or the composer got inspired by looking into a phage genome. Is that true?
Yeah, so we had a very fun project where there was a composer at the school, and he had a class, and they had to learn to compose as part of the class. And we gave them genes from a genome of a phage. So every student had its their gene, which had to be the basis of their composition. And they did amazing.
Can one listen to the music somewhere on
Yes.
YouTube or
It's on
the internet?
Yeah, it is. It's called Phage2015.org.
Okay.
Yeah.
Wow.
And there's the compositions, and it's visually beautiful too, because they wrote out the music, so you have the big printouts of what the music looked like, and then people, of course, did their style. So you have everything from hip hop to like a very classical sound to it. And some of the musicians were really creative, where it wasn't that they just resampled and made the music out of it. We had one guy who kind of reinvented the genetic code to make the notes.
Okay.
So it reinterpreted the whole genetic code. It was beautiful.
Oh, wonderful. Yeah. Okay.
Yeah, musicians actually have a pretty good feel, because I don't know, have you ever just listened to DNA? Have you ever?
No.
It's got this, you can actually hear the genes. If you just assign notes to the different base pairs, you can tell where a gene is versus a repetitive sequence. It's really cool.
Yeah. Oh, fantastic. Yeah. It seems we really live in exciting times, and so both in science, but also beyond the direct sciences. And I thank you very much for coming here, and we are very much looking forward to your talk.
All right. Thank you for having me. I appreciate it.